Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. Before we dive into this episode, we would like to take a moment and thank our ecosystem partners for their support, namely Domain Therapeutics, GPCR Therapeutics, Design Pharmaceuticals, Montana Molecular, and Orion Biotechnology. Mark your calendars for July 21st, as we will be hosting another free symposium in the ecosystem, this time on the molecular and structural insights of GPCR activation. Our program includes talks from Katerina Nemec, Alexei Serbo, and Polina Isankina. We're taking you on a structural and molecular journey to understand GPCR activation better. If you'd like to give a talk, we are still constructing the program, so please reach out by email at hello@drgpcr.com. We also set aside 2 hours for poster presentations and networking on a new space called Kumo Space for this symposium. There is no limit on the number of posters we can accommodate, and everyone is welcome to present a poster at our upcoming events, including the one on July 21st. We will not be selecting posters. You will not have to send in any posters. We only ask you to fill out the poster form so that we can capture your information. Part of that information is a 1-minute video abstract that we will share on YouTube. We'll also share it on our website so that people can see what you will be presenting about. You can also join us live for any of our events by marking your calendar and becoming a Dr. GPCR Ecosystem free site member. For more information and an updated schedule, you can go into the ecosystem. If you haven't noticed yet, we've added menu buttons at the top of our page for your convenience to help you navigate the ecosystem more effectively. And now, let's dive into this episode. Hello, everyone. This is Yamina from Dr. GPCR. It is not that early as it typically is when we record these podcast episodes, although I do feel like I need a second coffee. I'm very excited today to have with me today Tanishka Saraf. Hi, Tanishka. Super excited to have you on the podcast.
Thank you so much, Yamina, for this opportunity, and it's really an honor to be here.
Super excited to have you on. You and I interacted very briefly at the Dr. GPCR Summit. I think you were at the career panel, and I have to commend you to say amazing for you that you reached out to some of the panelists who came back and said, oh, she did reach out and we had a great conversation. So kudos to you. How about we start at the beginning?
Thank you to you, actually.
Of course, of course. So how about we start at the beginning and have you introduce yourself, and then let's get the party started.
Sure. So my name is Tanishka Saraf, and I'm from Mumbai, which is on the west coast of India. And I graduated in May 2018 with a Bachelor of Pharmacy from Institute of Chemical Technology. And shortly thereafter, in fall 2018, I joined Mercer University to pursue my PhD in pharmaceutical sciences in Dr. Clinton Canal's lab. And so I'm here in Atlanta now. And for my PhD, I've been studying serotonin receptors. As you know, like serotonin receptors, all of them except for 5-HT3, they are a family of GPCRs. And I have studied them in the context of a neurodevelopmental disorder, which is called Fragile X syndrome. It is the leading monogenic cause of autism spectrum disorder and intellectual disability.
Amazing. One of my questions typically is, did you always know you wanted to be a scientist? But before that, I want to ask you, how was the transition from Mumbai to Atlanta for you?
So I think it was really different. I mean, everything is different in Mumbai compared to here. Like not just climate-wise, but I don't know, like university-wise also things are a little bit different. But yeah, I think it was made easy by my advisor and my lab mates, because they really they made me feel like very much at home. And I think they really guided me very well through this process. So I'm really grateful to them.
Well, that's amazing. And it's so interesting because until you leave your comfort zone, you don't know what you're capable of. And then when you do, although it's scary, you realize that a lot of people are there to help you, lab mates, the PI, and then I think a year in, typically, you're already fully accustomed to your new environment.
Yeah.
So let's go back to the previous question. Did you always know you wanted to be a scientist?
No, actually, I did not, but I was really interested in science since when I was in school. And both my parents are actually commercial artists. So I was like always in a dilemma, like whether I want to pursue art or whether I want to pursue science, because I really like studying science. And I think my mom, she really encouraged me to pursue like whatever I was interested in, as opposed to like something that I would be supported for by them. And there used to be these young scientist competitions in India, and one of the notable ones was Homi Bhabha, which was named after the Indian nuclear physicist. And those exams they had like levels to them. So in middle school and high school, we used to take those exams. And then one day I came home and then I told my mom that this is like I've qualified for the next round. And but like I'm the only girl there. There are all boys like from my school. I'm the only girl. And then she just looked at me and she said that then you should really take this seriously, because you should value this opportunity, because you're the only girl from your school. And so I think that was like one of the defining moments for me. The other one I would say is, and this is like a very personal thing. So my maternal grandfather, he got diagnosed with Parkinson's disease when I was like in sixth or seventh grade. And later he also he got diagnosed with dementia, and the progression was like really, really fast. And I still like remember the episodes where he had like tremor or some aggressive outbursts, or just like a lot of dip in energy, and then a sudden burst of energy once he took his medication. So it was just a lot that the family was going through at that point. And also like medication wise, we had to pre-order and import some of his medication from the United States, because at that time like it was not readily available in India. Also like physician visits became like almost impossible after a certain time. So we had to do like phone consulting, and there was just a lot going on, and it was like a full gloomy decade of like no hope. And also it was very financially draining for my family as well. So that's when like I thought that, yeah, I mean, this is something really interesting, that we have literally like conquered the world. We have gone to the moon, and we still cannot understand what our brain is. So I think I was really that really piqued my interest in neurological disorders. And so that's where I decided that yes, I want to do something in this field.
Wow, thanks for sharing that. That must have been very intense. And I think based on what you're saying, it was definitely life defining, and who wouldn't find that life defining? Wow. And then you were interested in science, obviously. We now know why, but what made you interested in pursuing a PhD in the US?
So when I was an undergrad, I did take up an internship at a pharmacology lab, and they were working on Parkinson's disease, and they were just developing their zebrafish model. And so I was involved in a small part in that project, and I think I just learned like a lot of techniques, and I learned like how to read papers, how to critically evaluate results, and then how to plan an experiment. So I think all that really interested me in the research side of things. And then opportunity wise, in terms of the field of neuropharmacology, I think a lot more resources are made available in the United States. And I spoke to some of my some of the alumni from our undergrad college, and they were here in the United States, and they just told me like the state of art facilities that are available here to pursue higher education, and so that's how I decided to come here and pursue my PhD.
Wow. How did you pick your lab, knowing that you're very interested in
Hmm.
neuropharmacology, and then you knew that you wanted to come to the US? And the reason I'm asking is for all those students who are thinking of doing a PhD, how did you
Hmm.
go about picking the current lab you're in?
Hmm. So I was mainly looking at just labs that did that focused on neurological disorders that were either neurodevelopmental or neurodegenerative. I was not really looking at addiction studies. I I was not interested in those studies at that time. But yeah, so I was applying to labs. I was also looking at the different labs and the different facilities that were available at that university. Also, like in Georgia, like we have this alliance of universities, so I can avail resources from Emory or Georgia Tech if I need to. So yeah, all in all, I was looking at the research that was going on in the lab, the facilities, and then also like location was another important thing for me. So I don't like the cold. I was kind of trying to pick something that's warmer. But yeah, so finally, I think I did get accepted from three different labs in like three different locations. And then I had an interview with all of the PIs at all the institutions. And I thought that Dr. Kanal he gave me a lot of time. And I think the conversation that we had was really good. And I really had a lot of confidence after talking to him.
That's amazing. And I think that's very important, doing your due diligence, figuring once you know what you want, finding the place and the people and the university, the tools that you want to use, finding, really doing your homework basically based
Hmm.
on what do you want. And there's two elements in there. What do you want and
Hmm.
what is available out there? And it's the intersection of those, both of those things
Hmm.
that get you to the lab and to do the research that you're interested in.
And I think one more thing was that I wanted to learn how to work with animals. So that was another point for me. Like I wanted to do in vivo studies. So I chose something that had more in vivo component to it rather
Mhm.
than an in vitro only component.
Wow, amazing. It's so amazing how you knew what you wanted and you went after it and found the lab that and the university that gave you all the tools that you need to pursue your career. So where do
Mhm.
GPCRs come into the picture in your career, or when?
So I think the first time that I heard about GPCRs was in undergrad. We had a class where we touched upon like what are the types of receptors, and then in terms of GPCRs, we learned about like the signaling through GS, GI, and GQ couple receptors. We were actually studying, I think, adrenergic receptors, and I just vaguely remember this presentation slide that the professor had on, and it had a cartoon of a bear running behind a woman, and then oh, what's the fight or flight response, and and like what happens to the lungs in this case, what happens to the heart. So um, yeah, it's funny, but I remember it.
I
That
can
was.
imagine it, and it was very you cannot forget that because and it goes to the point that you were making, that you're interested in vivo effects, you're interested in global effects, and
Mhm.
the explanation was around what happens in these different organs. My
Mhm.
thought would have been what happens in the cell on a molecular level, but you're looking at it one
Uh-huh.
one level higher, which really,
Yeah.
you know, attests to the fact that you're interested in vivo effects, you're interested in organ effects and global effects on the organism.
Yes. So I think my training in undergrad was from like a very patient perspective since it was a
Mhm.
Bachelor of Pharmacy.
Yeah,
So I think I was always more interested in what the clinical outcomes would be of targeting these receptors.
that's
So yeah, that was, I think, sorry, go ahead.
No. There is a slight delay, so it always feel like we're interrupting each other, but we're not. Please go ahead.
Okay. So yeah, that was like the first instance where I even heard about what a GPCR was. And then when I came to the United States and I joined this lab, of course, like in lab meetings, we did talk about GPCRs, and then we also used to have joint lab meetings with another molecular pharmacology lab, and they also were studying adrenergic beta 2 and free fatty acid receptors. So I think like through those lab meetings, I learned a lot about GPCRs.
And you mentioned a little bit more about your project. Now, where do
Mhm.
GPCRs, or what is the role that GPCRs play in the project that you've been working on, or the different projects that you've been working on in the lab?
Mhm. Yeah, so I have been working on serotonin receptors, and I have been working on serotonin 2C, 2A, serotonin 7, and serotonin 1A receptors. So all of these are GPCRs. So 2C, 2A, they couple to GQ, 7 couples to GS, and then 1A couples to GI. So I have narrowed down, like obviously I started with all these receptors, but now I have narrowed down to 5-HT1A receptor. And we are looking at whether the inhibitory effects that this receptor has downstream, can they correct the neuronal hyperexcitability that's going on in Fragile X syndrome? So I can just give you like a background about what
Please.
is Fragile X syndrome. So it's a neurodevelopmental disorder, and it's caused by a CGG repeat expansion beyond 200 repeats in the 5' upstream promoter region of a gene called FMR1. And this essentially leads to like less production of the protein FMRP, and FMRP's inherent role is to repress translation of other proteins. So it regulates the translation of over 800 mRNAs. And so, in the absence of this protein, obviously the repression of translation of other proteins, it goes like unchecked. And so there's like a lot more upregulation in proteins. And so that's what causes like different ion channel expressions to go up, or even like different proteins such as excitatory like glutamate receptors, or like a decrease in functioning of the GABA receptors. So that causes like an overall hyperexcitability, and that leads to symptoms like intellectual disability, like learning and memory deficits, language delay, also like anxiety, hypersensitivity to external stimuli, seizures. So there is a like a plethora of neuropsychiatric symptoms that's going on. And so what we are trying to do is somehow, if we could modulate this excitability by targeting 5-HT1A receptors, then could we actually rescue some of these phenotypes?
Mhm. Wow. I think that's really phenomenal, because my next question, and you just answered it by the way, was gonna be, okay, so how do you, since it's genetic and you're inhibiting the production of this protein that regulates transcription of a plethora of genes, pharmacologically, how can you modulate, or at least play around with the phenotype to try and regulate that? But you did answer the question. And what does your day look like typically? Because you mentioned working with animal models. How do you apply this project, or how do you use animal models in the context of this project?
Right. So what we're looking at is the expression and function of 5-HT1A receptors in Fragile X mice as compared to the wild type control mice that we have. And of course, like we use tools like saturation binding assay or autoradiography, and also in vivo behavioral assays or electroencephalography. And using these tools, we first try to understand like what's the endogenous expression, or is there any like changes in conformation, or are there any upregulation, downregulation in the function? And then we are trying to therapeutically activate these receptors for treating a robust phenotype, which is audiogenic seizures in these mice. So audiogenic seizures is essentially because these mice are hypersensitive to sounds. If you expose them to a loud alarm, then they have a seizure and they might die. So we have actually seen promising results with activating these receptors, wherein we have been able to like dose dependently reduce the seizures. But yeah, I think that's like one, two, three parts of the project.
Yeah.
And there are just so many aspects to 5-HT1A receptors, like they're presynaptic, postsynaptic. So I think that's like the next few questions that we're trying to answer.
Mm-hmm. And how did you mentioned starting out with more serotonin receptors? How did you narrow it down to the one that you're working on with?
Right. So initially we started with 5-HT2C receptors, and there were reports of lorcaserin, which is a 5-HT2C agonist, that rescued like seizures in childhood epilepsies,
Mm-hmm.
in preclinically as well as clinically. So we first thought that okay, maybe like this could work in terms of Fragile X syndrome also, because it's like a neurodevelopmental disorder, and they also have seizures. And there was also evidence that 5-HT2C knockout mice, they have these audiogenic seizures, and they also have like some social deficits. So this was all like fitting in very well.
Yeah.
And so our hypothesis was strong, but we did not get the results that we desired. So we kind of ruled out that target. We also looked at like 5-HT2C and 2A expression and function, but we didn't really observe any like significant alterations in those. So then the next thing we moved to, excuse me, was 5-HT7 receptors. And so 5-HT7 receptors, there was a report that was published that they can actually help in learning and memory processes, but then they're also coupled to GS. So like will they increase excitability? So again, we just started first with looking at the expression of these receptors, and we didn't really see any alterations again. And in fact, the 5-HT7 receptor levels are really low in the brain. So then we moved on to 5-HT1A receptors, which is an inhibitory receptor. And simultaneously, there was another project that was going on in the lab, and that involved like a novel serotonin modulator, which is FPT. So my lab mate was working on that project at that time, and she observed that that compound, it was a partial agonist at 1A. It was very potent, high efficacy, and it was a partial agonist at 7, very potent, but again, like the efficacy was not very high, so it was almost like an antagonist. And she did observe that at a dose of 5.6 mg per kg, it blocked seizures in 100% of the mice that were tested.
Wow.
So that was also kind of like a result that pointed in a direction towards targeting 5-HT1A. And so I also like tested that compound in vivo for electroencephalography, and it did modulate like different frequencies and the strength or the power in different frequency bands. So, and this was also very promising because other drugs that are in preclinical or clinical trials have also shown these kind of modulations in like the overall relative power spectrum of EEG. So I think we just got like a boost in a positive direction towards pursuing 1A as the receptor target.
Wow, it's so interesting because I think it's a fascinating story. And in the background, there is a lot of communication, a lot of collaboration that goes on. Because if your colleague would have not communicated, you guys didn't have a chance to communicate, then you'd be potentially working on a different project or on a different serotonin receptor, or still looking for the one where you can see a modulation, which I think it's just phenomenal. Having worked with all these serotonin receptors, what is your favorite GPCR? which is a tough one. And it can be
Well,
more than one. It can be a family of receptors. It can be,
mhm,
I think Andrew Tobin had a favorite phosphorylation site.
mhm.
All
Wow.
answers are acceptable.
So I want to say it's 5-1A because it's giving me the results. No, I'm just kidding. So it was the first discovered GPCR in the serotonin family, and it also has like abundant expression all over the brain, like especially in the neural systems that are disordered in Fragile X syndrome.
Yeah.
And also, like, there is this added complexity and multidimensionality to this receptor where, it's present postsynaptically, it's present presynaptically, it's present as autoreceptors, it's present as heteroreceptors, and it's also present on like different types of cells. So it's present on glutamatergic, GABAergic, cholinergic, as well as serotonergic cells. And yeah, like it modulates the output of these cells, and they can also form like heterodimers with 5-HT7 receptors. And so I think just this level of complexity and multidimensionality that this receptor has, it makes it interesting to study it. And it also makes it interesting or challenging to use it for therapeutic purposes because you don't know what off-target effects it might have since it's so abundant in the brain. So, but nevertheless, I mean, there are drugs out there on the market that do target these receptors and that have been used for many clinical indications. So, yeah,
That was gonna
I
be
think.
exactly my comment, knowing that it's so widely expressed and it can form
Mhm,
dimers, heterodimers, and homodimers. And then that
mhm,
was gonna be exactly my comment, is how easy is it to target this receptor, knowing or not knowing completely the extent
mhm,
of its function in the brain, depending on the
mhm,
region and on the cell type it is expressed on.
mhm,
But it's so funny because sometimes you think about these GPCRs and you tell yourself, oh, we just need to hit this GPCR and induce this signaling pathway, and then it gets expressed on all sorts of cells. We don't understand what it does in all those cells, yet there are drugs in the clinic or drugs approved that target that receptor. And I think it's
mhm.
just miraculous these things
Yeah,
actually work,
yeah.
which is absolutely phenomenal. What are some of the other um, disorders or diseases that this receptor is implicated in?
Mhm. So, anxiety and depression are the two disorders in which it has been like prescribed for. So Buspar, which is buspirone, partial agonist at 5-HT1A receptors, it has been used as an anxiolytic. And there's aripiprazole, brexpiprazole. These are atypical antipsychotics, but they have also been used for depression and bipolar disorder.
Mhm.
There is uh, right now, NLX-112, which is a selective agonist at 5-HT1A receptors. It's in clinical trials for Parkinson's disease. They're also trying to test it for chronic pain, for neurodevelopmental disorders like Rett syndrome, which is another like neurodevelopmental disorder in which it's more prevalent in females as
Mhm.
compared to males, and that's like opposite of the prevalence seen in Fragile X syndrome. And I think just like learning and memory disorders or even seizure disorders, um, they also target like 5-HT1A receptors. Uh, a- Another like clinical indication is like irritability in autism. So aripiprazole is one of the only two drugs, aripiprazole and risperidone. They have been used clinically, and they have been prescribed for irritability and to manage these some of the behavioral symptoms in autism. And there was also a study that was published for Fragile X syndrome where it did show significant improvement in irritable behavior when it was given to children. So
Wow.
yeah, I mean there are a lot of indications for which there are drugs on the market, as well as like there are some in clinical and preclinical trials. But yeah, it's a wide range of disorders.
Wow. This means that you're working on a hot target and you have a lot of tool compounds to be studying this hot target, which takes me to my next question. And 100% of the time, the answer is yes. And I think just by you describing all the diseases and syndromes that your receptor is implicated in, the answer is definitely yes. But I'm still going to ask that, do you think that GPCRs are still good drug targets?
Yes, definitely. So, because they work in clinical settings, and I think that they are very well classified, like structurally or in terms of distribution, cellular localization, or the signal transduction pathways. And I just think that with the advent of like more selective compounds that target like one receptor subtype over another in the same family of receptors, or even like that prefer one signaling pathway over another, I think it could help a lot to reduce some of these adverse off-target side effects that are seen with traditional compounds.
I think so too. And I think worst case, they're going to be great tool compounds and allow us to really categorize what receptor does what. We'll never get the full picture because you can't look at the full organ at the same. It's hard, but at least it gives you an idea. And it's funny because I was talking to someone last week and we were just talking about the fact that as humans, we have this need of classifying and categorizing. So we categorize the GPCRs, for example, in different classes. But I wonder if anyone, and I don't know the answer to this, if anyone has thought about revisiting GPCRs in general based on their expression or based on the ligands that they might bind, and see, can we repurpose or recategorize or look at the different categories in a different way, other than, for example, sequence similarity or homology.
That's very interesting thought.
I think because, so the reason being is that we're focusing on one thing. We know that it's class A, and then this is class A, this is what typically happens, period. But have we thought about outside the box? If we didn't know it was a class A, if we just knew it's expressed in a specific cell and it controls these physiological events, would we be thinking about the class, or how would we approach drug discovery if we took out the already known classification components of it? I don't know. It's a rhetorical question. I don't know, but I thought it would be a really great thought exercise to
definitely
think about what would happen if we could do that. All right, super. Last segment, you talked a little bit about what drove you to become a scientist. Are there any other aha moments in your life, in your career as a scientist, that got you to where you are today?
Yes. So I think one of them is, I started with this, so I want to say this. So one was just like microscopy and learning like about the structure of the brain, and just looking at histological evidence or looking at the slides that you have seen, and just looking at, like I see these neurons here, I see these dendrites, I see these nets that are forming around the neurons, or I see these proteins that are inside the neurons. So I think the ability to like detect at that level, I think that was one of the things that I was like pleasantly surprised about, and I was like, wow, yeah, this is great. And the other thing is, I think when I started working with electroencephalography, I think just the fact that you can measure brain waves, and you can understand that there is like an electrical language of the brain by which like these different regions communicate with each other, and how they respond to external stimuli, or even how they process information. I think that that was a really great moment for
Yeah.
me to just see it live, and also like how these brainwaves are impacted once you treat the animal with a xenobiotic. So what is the change in the frequency or the power in the frequency? And so I think you can extract a lot of information from a live animal, like without having to euthanize the animal. So you can study the brain, while studying behavior or while looking at information processing. So I think that was also one of the moments where I felt like, wow, this is great.
It's interesting
And
because, you know,
there's one more
it's,
thing, actually. Sorry,
no,
I
no.
keep saying this like to like one of my junior lab mates, but I always tell her like, oh, the dose is what differentiates a drug from a poison. And it's not necessarily an aha moment, but I just like that statement.
Yes.
It's I think it's by Paracelsus. He was
I
a
agree.
Renaissance physician.
Mhm.
Yeah, but I don't know. I just really like that statement, and I think it's a good statement to remember.
Yeah. Yeah, no, I think obviously all true. And it's so interesting because all the examples that you gave are about seeing something and
Hmm.
experiencing something. And I feel like whenever we're thinking about molecular pharmacology or pharmacology, you see the consequence in a test
Hmm.
tube, or you're measuring the consequence in a plate reader, but being able to see it, we know it's there, but being able to experience it and see it happening, I think that just gives it an additional, you know, um, proof of reality that it does exist,
Hmm.
which
Yeah,
I find absolutely amazing.
yeah.
I know you're finishing up, where you're in your PhD program. Do you have any advice for scientists who want to get into a PhD or are currently doing a PhD, something that you've learned and you want to pass on?
Yeah, sure. Yes. So I was very shy in asking for help, like early on in my PhD, and I thought, oh, like this is a stupid question. I don't think I should ask this in front of so many people.
Yeah.
But I think that that's very wrong. And I think that if you're starting early on, then it's okay to ask for some help. And you don't have to reinvent the wheel. It's fine to ask for advice, ask for help. And it's okay to be standing on the shoulders of giants to look for your answers. So, um, yeah, I think that it also saves you a lot of time instead of you like going through so many things like the whole
Yeah.
literature. And I think that I realized this when the pandemic hit, because I was excited to go to conferences, and then all of a sudden there were no conferences. And so I thought that, oh, like when am I going to meet people? When am I going to talk to them?
Yeah.
And I think that's when I realized that, yeah, I mean, I should take up every opportunity that I get to, connect with people who are in my field, and just talk to them in general.
Yeah.
How they're like, how is it going for them? And yeah, I think you just realize after a point that like everybody's in the same boat. So um, it's
Yeah.
okay to ask for help.
I think you're making a phenomenal and very important point. It is okay to ask for help, and it is smart to ask for help,
Hmm.
because as you mentioned, you're not there to reinvent the wheel. You're there to learn how to think.
Hmm.
And no matter, even if you're a postdoc or even if you're a PI, it's okay to ask for help. Nobody knows everything. And I think
Hmm.
putting a lot of, putting your brain together with other people's brains, it just exponentially enhances our ability to comprehend science. And
Hmm.
it's not a solopreneurship when you do science.
Yeah.
Nobody can do it on their own. I mean, if they can, good for them, but I don't think that's even possible,
Hmm.
because it's so complex. Like you mentioned in the beginning that, we've went to the moon, we're
Hmm.
launching rockets, but then we still don't understand exactly and fully what happens in the brain. And we need to
Hmm.
put our brains together in order to advanced science. And I think it's sound advice when it comes to asking for help.
Mhm.
I think it can also be way more valuable to know who to talk to than
Yeah.
figuring out the answer, because sometimes you may have a question and you may think one way about the question, but having a conversation with someone. And I like to talk to two types of people: experts in that
Mhm.
particular field and non-experts.
Mhm.
Because the expert
Right.
is going to point you to the paper, to the explanation, to the history. They know this by heart. The non-expert might
Mhm.
ask you questions that
Mhm.
make you think and
Yeah.
really
Yeah.
approach the problem from a different angle.
Yeah. Yeah. I agree.
So that's definitely something important. Any other advice that you can think of? If not, I have one last question for you.
Uh, yeah. I can't think of anything right now.
I think one thing that you're kind of the living proof of it is the fact that you moved from your country to a new one that
Mhm.
culturally is different, that is absolutely different, but
Mhm.
then you did it in a smart way, because first you figured out what you wanted to do, and then
Mhm.
you found out where you wanted to do it. And I think that's something that
Yeah.
people can learn from. And kudos to
Mhm.
you for being determined and knowing what you wanted and then going after it.
Thank
My
you.
la- my last question is, and you may say, Yamina, I have no idea. My last question is, where do you see yourself after your PhD?
So right now, I'm just open to any opportunity that comes my way,
Mhm.
but I would still like to be in the neuropharmacology field.
Yeah.
I would still like to work on neuropsychiatric disorders, and yeah, like uh, mostly like something that can help patients.
Mhm.
Um, so yeah, I I think I see myself in a role like that.
Mhm. Would you see yourself continuing to work in the lab, or maybe potentially lead a team?
Yeah, I think my advisor always like pushes me to like push my boundaries.
Yeah.
So I think yes, like I have learned a lot of lab work, but I have also understood like how to do experiments or how to plan a project. And so I
Yeah.
think I do see myself in a more of a leadership role. Of
Mhm.
course, like I don't mind being on the bench. I don't necessarily hate lab work. I do like it.
Mhm.
But yeah, I mean, I would be really honored if I were to
Yeah.
be in a position where I were to lead a team.
Mhm. And I think that's the natural evolution of things,
Hmm.
because at some point you become such an expert at the
Hmm.
bench work, the lab work, and um, then you grow into a role where you can learn new things and then help
Hmm.
people grow into their lab position and learn their technical skills and guide those
Yeah,
people.
yeah.
So I think that's kind of a natural evolution of things.
Hmm.
Super. Thank you, Tanishka, for being here. I really appreciated our chat, and good luck with everything. You mentioned you started your PhD in 2018,
Yes,
so you're soon graduating, I assume then.
yes. Hopefully by the end of this year.
Well, good luck with that. Let us know if we can help with anything, and let's keep in touch.
Yes. Thank you so much, Yamina.
Thank you so much for being here. And now we're going to let people wonder what we're going to be talking about after we stop recording. I've been playing this joke for quite some time now. I think I need to renew renew the joke. All right. Thanks.
Bye.
Thank you for joining us and listening to this Dr. GPCR podcast episode. We would like to thank our guest. We also want to say thank you to our Dr. GPCR team members: Attila, Ines, Montserrat, Ivana, Andreina, Balint, and Julia. A huge thank you to our ecosystem partners as well: Domain Therapeutics, GPCR Therapeutics, Design Pharmaceuticals, Montana Molecular, and Orion Biotechnology. You can connect with our partners directly in the ecosystem, so don't forget to join us today. Please subscribe to the Dr. GPCR newsletter, find us on YouTube, and if you like our podcast, leave us a 5-star review on Apple Podcast. You can also leave us a testimonial at drgpcr.com/testimonials. Another great way to support us is to share your favorite Dr. GPCR program with your network and colleagues. With any questions or suggestions, feel free to email us at any time at hello@drgpcr.com. Until next time, stay safe.